Good morning. Every year, the Society for Applied Microbiology awards the W.H. Pearce Prize, our most prestigious award, to an early career microbiologist who has made substantial contributions to the science of applied microbiology. Each year, our members nominate individuals across all sectors of applied microbiology who they deem to have conducted outstanding original research or made outstanding contributions to the field. I'm delighted to say that the W.H. Pearce Prize for 2020 has been awarded to Dr. Joan Geoghegan, Senior Lecturer in Microbiology and Infection at the Institute of Microbiology and Infection at the University of Birmingham and formerly Associate Professor of Microbiology at Trinity College Dublin. The W.H. Pearce Prize was instituted in 1984 by the directors of Oxford to commemorate the life and work of the late W.H. Pearce, a former chief bacteriologist of OXO and a longtime member of the Society. We are grateful to the Pearce family and to Thermo Fisher, who generously sponsor this award. Indeed, the Society's links to the Pearce family remain strong. I'm delighted to say that Bill Pearce is the grandfather of Nicola Williams, member of our early career scientist committee, and their amazing artwork regularly adorns the cover of our Society magazine, The Microbiologist. Nominations for the 2020 W.H. Pearce Prize are now open, and I would encourage members of the Society to nominate colleagues who they feel have made outstanding contributions to the field. The W.H. Pearce Prize celebrates and amplifies the outstanding work of our colleagues at an early stage of their career. The link to nominate is on our website under Awards and Prizes. But back to our awardee for 2020. This award celebrates the outstanding research of Dr. Joan Geoghegan, whose research focuses on understanding the molecular basis of colonization and infection by Staphylococcus aureus, identification and characterization of bacterial factors that promote adhesion, biofilm formation, and immune evasion. Her PhD in 2009 involved the biochemical and biophysical characterization of proteins expressed on the surface of Staph aureus under the supervision of Tim Foster at Trinity College Dublin and Magnus Hook at Texas A&M, leading to a redefinition of the fibrinogen binding site of the major Staph aureus fibrinogen binding protein, and identification of vaccine candidate antigens as part of an exclusive license with GSK. As a postdoctoral researcher at both Trinity College Dublin and University Medical Centre Utrecht, Dr. Geoghegan initiated early studies on the molecular basis of protein-mediated biofilm accumulation in Staph aureus. Her work dissects the mechanistic basis of adhesion to host surfaces and biofilm formation in MRSA to discover conserved interactions that can be targeted for therapeutic agents. Her research investigates the individual protein interactions that are crucial for building and maintaining a biofilm in contemporary MRSA isolates. Understanding these interactions has allowed her to identify blocking molecules that interfere with early stages of biofilm formation. Joan has an international reputation for outstanding research in this field, leading to a number of international studies on biofilm formation and colonization. She has published her research widely in leading journals where her contributions have been highly cited. So, on behalf of the Society for Applied Microbiology, I congratulate Dr. Joan Geoghegan on her outstanding research and the award of the W.H. Pearce Prize 2020. Joan is unable to join us for the Q&A after the presentation as she is currently on maternity leave. So again, we extend our warmest congratulations to Joan. I now invite Joan to deliver her W.H. Pearce Prize Lecture 2020 entitled Host Colonization by Staphylococcus aureus, Cell Wall Anchored Proteins at the Helm.
Good morning, everyone. I'd like to begin today by thanking the Society for Applied Microbiology for this award. I'm delighted to be able to deliver this talk on the cell wall anchored proteins of Staphylococcus aureus, And I hope that this will provide a sense of how cell wall anchor proteins are really key for this bacterium to successfully recognize the human host and to establish itself within a host niche through adherence. So my group are primarily interested in understanding the molecular basis of Staphylococcus aureus colonization and pathogenesis. And this gram-positive bacterium adopts a dual lifestyle, both as a commensal and as a pathogen. So it's estimated that up to 30% of the human population is persistently colonized by S. aureus, while the remainder of the population are intermittently colonized, meaning that they carry the bacterium transiently. The primary colonization site in humans is the anterior nares, and the majority of those who are colonized will carry the bacterium at this site. However, Staph aureus is also capable of colonizing the oropharynx, various regions of the skin, as well as the intestinal and rectal regions. So while Staph aureus is a commensal, we know that it is also a very important human pathogen. And being colonized by Staph aureus is a risk factor for the development of infection. Staph aureus causes a wide variety of different infections that range really from very superficial skin infections to much more serious, life-threatening invasive infections. And remarkably, as you can see here, this bacterium is capable of infecting almost every tissue and organ in the human body, leading to a variety of infection types, including pneumonia, infective endocarditis, osteomyelitis, and severe skin and soft tissue infections. As a leading human pathogen, S. aureus is extremely problematic due to high rates of hospital admissions and mortality. There are limited treatment options available due to the widespread antibiotic resistance, because Staph aureus has effectively evolved resistance against every antibiotic currently licensed for the therapy of staphylococcal infection. And despite what we've seen with the very rapid development of a vaccine to protect against COVID-19, there is still no effective vaccine to protect against Staph aureus infection on the market, despite more than 25 years of research in this area and efforts to develop an effective vaccine. So Staph aureus is clearly a very adept colonizer and pathogen, and one of the tools in its arsenal that allow the bacterium to be so effective in this context are the surface located cell wall anchored proteins. These proteins are anchored to peptidoglycan and extend through the cell envelope to the surface of the bacterium, where they are displayed and where they exert their activity. These proteins are linked to the pentaglycine crossbridge of peptidoglycan by the activity of enzymes known as sortases. In the case of Staph aureus, these are sortases A and sortase B. So in vivo models have very clearly shown that cell wall anchor proteins have a prominent effect on virulence and colonization. And therefore, or that is perhaps not surprising when we consider the multitude of properties that these proteins confer to the bacteria. So the activity that was first recognized with these proteins, and that they're perhaps best known for, is their ability to promote bacterial adhesion. Now, this helps Staph aureus to adhere to the extracellular matrix, to host cells, or to biomedical or biomaterial device surfaces that have become coated with host plasma proteins. And this is a very important way for Staph aureus to initiate colonization or infection. The cell wall anchored proteins also contribute to biofilm formation, allowing bacteria that are adherent to grow as multicellular communities in vivo. Attachment to host cells can often lead to internalization of bacteria, and S. aureus can actively invade endothelial and epithelial cells using a selection of cell wall anchor proteins that promote uptake. Interaction with host cell receptors can also lead to inflammation. Some of these cell wall anchored proteins have pro-inflammatory effects. And while I'm not going to discuss it today, cell wall anchored proteins are also known to play a role in immune evasion, and indeed in iron acquisition through their ability to interact with various serum proteins. So it's clear that the Staph aureus cell surface is covered with this extensive array of proteins that fulfill numerous functions at the interface between the bacterium and the host, and act as major players when it comes to colonization and infection. The collection of cell wall anchor proteins is classified into different categories based on conserved structural domains. And the family of cell wall anchor proteins that my group is particularly interested in are the MSCRAMMs. And this stands for microbial surface components, recognizing adhesive matrix molecules. There are 7 of these proteins produced by Staph aureus, and the defining feature of an MSCRAMM is the presence of adjacent immunoglobulin-like folds. And we'll see in a moment why these adjacent folds are so important. These domains in MSCRAMMs are known as N2 and N3. And the N2 and N3 domains are linked to the cell wall by flexible, unstructured repeat regions, either serine aspartate dipeptide repeats or fibronectin binding repeats in the case of FnBPA and FnBPB proteins. And these repeats clearly also have ligand binding activity. They interact with fibronectin. So the repeat regions link to the cell wall, where we have the cell wall anchoring domain with a characteristic LPXTG motif. Towards the N terminus is a region of unknown function known as N1. And then we have the signal peptide, which directs export through the sex system. And a number of years ago, my group also identified a short peptide region located at, between N1 and N2 that assists with secretion of these proteins. So let's take a look at an example of an MSCRAMM. And the example that I'm going to use here is of clumping factor B, abbreviated to CLFB. And this is a protein that we'll hear more about during this talk. CLFB allows me to illustrate that the adjacent Ig binding or Ig-like folded domains that define an MSCRAMM are crucially important to the bacteria and to the function of these proteins, as these form the ligand binding region. So we see here in cartoon form that the cell wall anchored region, unstructured repeat region, acting almost like a stalk, is projecting the N2, N3 ligand binding domain away from the surface of the bacterium. At the interface between the N2 and N3 subdomains is a hydrophobic trench. We can see this here. The top of the crystal structure is N2 and at the bottom is N3. The hydrophobic trench that's formed between these domains is the major ligand binding region that accommodates the target sequence within a host protein. And in the case of CLFB, this protein tends to bind to sequences that are very rich in glycine serine amino acids, such as the alpha chain of fibrinogen and the corneocyte proteins loricrin and cytokeratin 10. So I think that you can really clearly see from what I've told you that MSCRAMMs are centrally involved in the interaction with ligands. And it is the MSCRAMM ligand interaction that underlies the ability of Staph aureus to adhere to host molecules and to host cells. And as a way to illustrate this, I'm now going to tell you about our recent work that has been investigating the role of MSCRAMMs in bacterial adhesion to skin during colonization. And I'll show you how we were building a molecular understanding of the adhesive interactions governing the host pathogen with the host bacterium interaction. So this brings me to talk to you about the role of Staphylococcus aureus colonization. And I'm going to talk to you about an ongoing study where we are trying to understand how Staphylococcus aureus colonizes the skin of patients with atopic dermatitis, which incidentally is also known as AD or eczema. And in particular, we're trying to understand the role of adhesion in colonization. So you might wonder, why focus on atopic dermatitis? Well, it turns out that the skin of atopic dermatitis patients is extremely susceptible to Staph aureus colonization. It's estimated that as many as 90% of these patients carry S. aureus on their skin. And this is in contrast to normal healthy skin, where colonization with the bacterium is transient. Atopic dermatitis is itself a chronic inflammatory skin disorder that is characterized by the presence of a dry, red, itchy rash and a disrupted skin barrier function. This can lead to severe discomfort for the patient and a reduction in the quality of life. Between 15 and 30% of children worldwide suffer from atopic dermatitis, and this disease can also carry on into adulthood, albeit with a much reduced incidence. So we know that the skin of patients is highly susceptible to Staph aureus, and unfortunately this has some adverse outcomes. Staphylococcus aureus can influence the progression and severity of atopic dermatitis. What happens is that the bacteria adhere to the skin, exploiting the weakened barrier function and indeed worsening the skin barrier function as they drive the inflammation that is so closely associated with atopic dermatitis. So in summary, Staphylococcus aureus colonization is problematic in these patients as it leads to the exacerbation of atopic dermatitis symptoms. So the very first step in colonization is often adhesion. And in the study carried out by Don Leung's group in 2001, gave us the very first indication that Staphylococcus aureus might display increased adhesion to skin affected by atopic dermatitis. What we see here are skin biopsies that have been incubated with S. aureus. On the right of the slide, we see normal healthy skin. And there are very few bacteria adherent to these biopsies, while on the left of the slide, we see a biopsy taken from a patient with atopic dermatitis. And here we can clearly see that a large number of bacteria stained in purple have adhered to the biopsy sample. And what's interesting about this is that the majority of the bacteria are adherent to the upper layers of the epidermis, a region that is known as the stratum corneum. So it seems that the colonization is mostly confined to the stratum corneum, with very little penetration of the bacteria to the lower layers of the skin. The cells that are located within the stratum corneum are known as corneocytes. These are essentially dead, anucleated, flattened cells that derive from keratinocytes that have undergone a process of terminal differentiation as they move from the proliferating layers of the epidermis to the outermost layer, the stratum corneum. The adherence of Staph aureus to the stratum corneum in AD suggested to us that the bacteria were adhering to the corneocytes. And this was very interesting because we know that corneocytes present in atopic dermatitis skin can be very different to the corneocytes present in normal healthy skin. One of the major factors contributing to this is natural moisturising factor, or NMF. And one of the properties of NMF is that it controls hydration of the stratum corneum. Loss of function mutations in a gene called filaggrin are quite common in AD patients, or there are also other causes of low NMF. But what we would generally say is that AD skin is often affected by low NMF, while normal healthy skin has much higher levels of natural moisturising factor. So whatever the cause, the consequence of low NMF is that the corneocytes adopt a different surface topography. This was very elegantly shown by Reitmuller et al. In 2015, who demonstrated using atomic force microscopy that the normally flat surface of corneocytes is covered with villus-like projections, or VPs, in atopic dermatitis. And we were later able to show that Staph aureus adheres better to low NMF corneocytes, that is, corneocytes that feature these VPs. We can see the villus-like projections here on the corneocyte surface. Then to high NMF or healthy corneocytes that, as we can see here, lack villus-like protrusions and have a very flat surface. And this told us that low NMF corneocytes that are typical in severe AD are better able to support Staphylococcus aureus adhesion. And of course, we wanted to understand the molecular basis of this. So in collaboration with Yves Dufresne's group at the Université Catholique de Louvain, we set out to investigate the S. aureus corneocyte interaction using an ex vivo model. And this involved tape stripping the AD patient's skin to isolate corneocytes. And then a single viable bacterium was linked to an AFM cantilever and used to scan the corneocyte surface. We used a representative clonal complex 1 strain that we call AD08 for this study. This strain was isolated from the infected skin lesion of a male patient with severe AD. Choosing a CC1 strain for our study was based on the work of Orla Fleury, who is a previous PhD student in the group. And she found that strains from the clonal complex 1 lineage were the most prevalent isolated from atopic dermatitis patients with severe to moderate disease. The results of our study very much agreed with similar studies carried out in other geographical locations, including Denmark, Spain, and Canada, where clonal complex 1 strains were also the most frequently recovered Staph aureus lineage in infected skin lesions. So with this clonal complex 1 strain, we used multiparametric imaging to map adhesion corneocyte interactions. And single cell force microscopy was used, and it involved immobilizing the single Staph aureus bacterium on the tip of the AFM cantilever, as we saw, and then measuring interactions between this functionalized probe and the corneocyte surface. So here in the top panels, we see the corneocyte topography, which has been mapped using AFM. And below, where the probe has been moved rapidly over the corneocyte surface to map any adhesion events. In the case of AD08, multiple adhesion events occurred, with each indicated with a colored pixel. However, when the mutant deficient in the MSCRAMM CLFB, which I introduced to you earlier, was probed, the number of adhesion events was greatly diminished, indicating that CLFB makes a major contribution to adhesion. Single cell force microscopy was then used to measure the force of binding of a single bacterium to a corneocyte. And you can see here from the average binding forces measured for the wild type and the mutant across 2 AD patients, that the loss of CLFB leads to a reduction in the binding force. Again, indicating to us that CLFB binds to low NMF corneocytes in AD. Establishing that CLFB promotes adhesion of Staph aureus to AD corneocytes led to a very important new question, and that is, what is the corneocyte ligand that this protein is recognizing? So let's come back to the topic of corneocyte topography. Corneocytes with low NMF and villus-like protrusions support more adhesion by Staphylococcus aureus than corneocytes with high NMF lacking VPs. So we hypothesized that the altered corneocyte surface might alter ligand presentation and bacterial adhesion in AD. Our attention was, of course, drawn to the VPs. Importantly, a previous study had shown that VPs are decorated with a protein known as corneodesmosin. Corneodesmosin is a cell adhesion protein, normally confined to the periphery of corneocytes and involved in cell-to-cell adhesion, as I said, and as shown here, when high NMF corneocytes were probed with an antibody raised against corneodesmosin. However, it turns out that in low NMF corneocytes, VPs themselves are decorated with corneodesmosin. Corneodesmosin sits at the tips of the VPs and therefore completely covers the corneocyte surface. And we believe that this might mean that the CDSN was far more accessible to bacteria. So the abundance and accessibility of CDSN led us to investigate if this protein serves as a ligand for S. aureus. And this was work carried out by Aisling Towell, who's a previous and recent PhD student in the group. So by purifying recombinant corneodesmosin, Aisling was very nicely able to show that the lab strain Newman of Staph aureus bound efficiently to immobilized corneodesmosin protein. And quite nicely, a mutant deficient in CLFB lost the ability to interact with corneodesmosin. So this told us that Staph aureus recognizes corneodesmosin and that CLFB is a major contributor. However, things changed when we looked at AD08, our clinical isolate of Staph aureus. Here we saw that there was no reduction in adhesion when a mutation in CLFB was introduced. So this was a little bit puzzling, but it really inspired us to look more broadly at MSCRAMMs to see if we could identify another MSCRAMM produced by AD08 that might be encouraging corneocyte adhesion. And interestingly, we found that one of the fibronectin binding MSCRAMMs, known as fnbB, also contributed to corneocyte adhesion because a mutant of CLFB was deficient. And interestingly, the very closely related fnbA gene had no effect on corneocyte adhesion. So we then decided to combine mutations to see if we could identify the full repertoire of corneodesmosin binding proteins. And quite nicely, a combination of CLFB mutation with the fnbB mutation led to a complete loss of adherence to corneodesmosin, while other combinations of mutations, including an fnbA, fnbB mutant, did not show this loss of corneodesmosin adhesion. So having discovered these two MSCRAMMs involved in binding to corneodesmosin, we took a closer look at the protein itself, the host binding partner. And corneodesmosin comprises a series of glycine and serine-rich regions at the N and the C terminus of the protein. Now, we already knew that CLFB likes to bind to GS-rich regions. So we decided to test the hypothesis that Staph aureus was recognizing either or both of these glycine-rich regions. So Ashing made in-frame deletions within the C-terminal or N-terminal region and studied the binding of the bacteria to these truncated proteins. What she found was that the deletion of the N-terminal region of the protein led to a complete loss of corneodesmosin adhesion. And indeed, expressing the N-terminal region alone supported Staph aureus adhesion. And taking these two pieces of evidence together, we were able to say that the N-terminal region of corneodesmosin is the part that supports Staph aureus adhesion. And interestingly, removing the C-terminal region had no effect on binding. So having mapped the corneodesmosin, or the Staph aureus binding site within corneodesmosin, to the N-terminal region, got us interested in this region, and we had an antibody that recognized specifically the N-terminus of corneodesmosin. And we used this antibody to see if we could block Staph aureus interaction with corneocytes. And we found that yes, this antibody could efficiently block Staph aureus adherence to low NMF corneocytes. We see this here. The antibody is reducing cell adhesion probability in a concentration-dependent manner. So this told us that Staph aureus interacts with the N-terminal region of corneodesmosin on the surface of patient corneocytes, which is very interesting. We also tested our mutants, CLFB and FnBP, for adherence to corneocytes. And as you can see from the data here, the loss of either fnbB or CLFB protein led to a 60% reduction in adherence. And we also see here there were fewer bacteria adherent to the corneocyte when we consider the mutants versus the amount of adherence in the wild type. So taking all of these results together, we can confidently say that Staph aureus interacts with the N-terminal region of corneodesmosin. The interaction with corneodesmosin is mediated by fnBPB and CLFB, and the binding to corneocytes is reduced when the N-terminal region of corneodesmosin was blocked with an antibody, indicating the contribution of this Staph aureus corneodesmosin interaction during bacterial adherence to human skin in atopic dermatitis. So in summary, I think what we can say is that while Staph aureus, or while MSCRAMMs have a plethora of different functions in Staph aureus, their activity as adhesins operating at this interface between the bacterium and the host is key to supporting virulence and colonization. This is really borne out by many studies from multiple groups over the years, and I'm very happy that my group have contributed to this knowledge, both in terms of the fundamental aspects of the MSCRAMM ligand interactions, but also in terms of characterizing interactions with in vivo relevance, as we heard about today with the example of the recent study uncovering the molecular basis of Staph aureus' interaction with host corneocytes in atopic dermatitis. So overall, we should consider these proteins as excellent candidates for interventions to prevent or treat Staph aureus infection, as if we can disrupt the adhesive protein-protein interactions, then we can interrupt colonization and infection. For example, if the CIFB and FnBPB interaction with corneodesmosin could be targeted, this would be an efficient way to reduce adherence to the stratum corneum in atopic dermatitis. So to finish this talk, I'd really like to acknowledge the many people who've contributed to the work in the lab over the years. Of course, the PhD students and postdocs who have worked within the group at Trinity College Dublin and more recently at the University of Birmingham, and these are the current group members here. And in particular, I'd like to thank the scientists who contributed to the research I've told you about today. They were Aisling Towell, Orla Fleury, and Tina Da Costa, and who worked on the atopic dermatitis skin projects. And I'd also like to thank the many collaborators on that project, especially Eve Dufresne, Alan Irvine, who were instrumental to all of the atopic dermatitis work. I've also had many other good collaborators and mentors over the years, particularly Tim Foster, Magnus Hook, Pietro Speziale, and Jennifer Potts, who all work in the field of MSCRAMMs, and many others who I've collaborated with on other projects that I just haven't had time to tell you about today. So I'm very grateful for the support from various funding agencies, and I'll finish by thanking everyone for this opportunity to speak today and for the award of this prize. And thank you very much for listening, and goodbye.
Thanks, Joan, for a fascinating lecture, and congratulations once again on your outstanding contributions to the field and the award of the 2020 WH Pearce Prize Lecture. Just to draw your attention once again to the fact that the WH Pearce Prize nominations for 2021 are now open, and you'll find the link to the nomination form for SFAM members at the bottom of this page. SFAM members will also be receiving email communication later on today about the nomination process. And I again encourage you to nominate outstanding colleagues for their contributions to the field for the WH Pearce Prize in 2021. We look forward to this afternoon's sessions on medical technologies in the fight against AMR and rapid antimicrobial susceptibility testing. We hope you can join us for those. If not, enjoy the rest of your afternoon and the rest of the conference. Thank you.